In this second half of the Biomedical Investigations Lecture 3 on genetic technologies, we're going to look at single nucleotide polymorphism analysis and also look at SNP chips, where we can perform hundreds of thousands of analyses on a single chip. So SNPs, or single nucleotide polymorphisms, are single base pair changes that are typically not pathogenic. And however, single nucleotide polymorphisms elsewhere on the genome can be pathogenic. Now, there's some great technology to analyse for single nucleotide polymorphism analysis, and these are offered by commercial companies such as 23andMe and Ancestry.com, and we'll call this consumer genomics. The technology behind that is offered as part of consumer genomics is also used in cancer subtyping. So I'm going to talk about how this technology works, and then we'll go on to more targeted single nucleotide polymorphism analysis. But basically these work by analysing about 700,000 regions of the genome where single nucleotide polymorphisms are known to exist, and these are analysed on a single SNP chip. A SNP chip is a glass slide impregnated with oligonucleotides which capture DNA and interrogate the genotype at each individual place. Now, most SNPs are silent changes. They don't really bear any resemblance to any pathology at all. A lot of them are non-coding, but some of the ones that can be assayed are pathological mutations. So this slide illustrates how the Illumina B chip works, and this is the one that's used by Ancestry.com, and it also can be used by people doing biomedical research, which is what it's actually designed for in the first place. So total human DNA is isolated and briefly amplified and generated as short sections of DNA. This is then applied to a bead chip containing 700,000 capture beads impregnated onto a glass slide. Each capture bead has a different oligonucleotide sequence attached to it, and this sequence represents a piece of the human genome just up to the point prior to where there is a known single nucleotide polymorphism. So this sequence here, if we look at the human genome, the next nucleotide is known to be a SNP that could either be a G or a C, for example. So what we do is allow fragments of human DNA to hybridize to these oligos. There we go, a couple of bits of DNA have stuck. And then once they've all hybridized, we supply the slide with labeled chain terminator nucleotides, labeled with different colors. So here comes the DNA polymerase and the nucleotides, and the nucleotides basically are added in. You add in just one nucleotide to extend the oligo on the glass chip using the template DNA as template. And what we can see here, the DNA that's stuck, there's a G there, so the next nucleotide we're going to add in is a C, which is red. And this piece of DNA, the next one that comes in, is going to be a G, because that's a C, that's a G, so that's going to be green. And the total light emitted from this is going to be a combination of red and green light, which is going to give a sort of an orangey. We'll be able to do spectral isolation of what colours are there. So, if green's incorporated, you get a G. If red and green's incorporated, it's heterozygous. And you can combine all of these four colours. And what you get is something that looks something like this, but this is a tiny fragment of it. And you get, for each individual spot, you get a colour. Some of them are green, some of them are red. A lot of them appear to the naked eye as being orangey. There's different combinations here. But from this, you can read this chip, the whole chip, this is just a tiny fragment of it, and then get the genotypes for all 700,000 locations. So this is how a SNP chip works. Now we can use this in lots of biomedical applications if we want to study a particular disease situation and ask the question which, if any of these single nucleotide polymorphisms correlate with disease, you can use these sorts of chips. And this link here takes you to the Illumina video explaining how they work in more detail. So that is one way to detect single nucleotide polymorphisms, and you can buy gene SNP chips like that are impregnated with pathological mutations and screen a whole load of mutations simultaneously all the way across the genome. What we can also do is assay for single nucleotide polymorphism or single nucleotide polymorphism changes using the principles of quantitative PCR. Now we would do this if we knew what we were looking for. We would do something like this if you wanted to go dredging for an association between one genetic change and a disease. You might say, well, let's look at all of the known variations and see if any of them just happen to correlate with our disease situation. So this is almost like more of a fishing exercise, whereas if you're using quantitative PCR, you need to know what you're looking for, and you're asking the question, is what I am looking for there? Because this particular phenotype can only be caused by one or two different changes. So for example, if we're interested in cancer subtyping, we may know that there's a mutation in an oncogene that drives cancer, and we may want to know whether that mutation is present, because that might determine what drugs we give. So we create PCR primers that amplify across the polymorphic or the mutation region, and then design a probe that matches either the normal wild-type allele or the mutant allele. It could be a G wild-type, it could be a T mutant. And then perform amplification and see which probes bind to the amplified DNA, and that will give you your genotype. And this is shown here. So when we do Typeman, this is using Typeman principles, and you should have learnt about Typeman principles earlier in the module. We perform PCR amplification across a piece of DNA, and there's a site in the middle here where this probe is binding where the polymorphism or mutation is. And this is just showing one strand of the DNA. Remember, the DNA is double-stranded. So here's a forward primer, here's one strand of DNA. We're not even showing the other strand of DNA, and we're not showing the reverse primer. So this PCR primer here is going to bind, DNA polymerase is going to extend in this direction, come across a probe in its path, and it will cleave off a reporter from a quencher. The quencher stops the reporter from fluorescing, but when DNA polymerase comes along and removes the quencher, this reporter can then fluoresce. And the amount of fluorescence from this reporter tells you how many copies, how many times DNA polymerase has found a probe blocking its path. So if that has occurred lots of times, then there is more template DNA being amplified. What we're doing here is using two different probes, two different colours, so the blue one and the orangey one. And this probe here recognises the wild type situation. So if we focus in the middle here, a homozygous wild type individual, only the wild type probe, well, the wild type probe would bind in preference. Now this probe sequence here is about 10 to 12 nucleotides in length, and if one of them is a mismatch, it doesn't bind particularly well. So if there's a single nucleotide polymorphism change, a wild type probe will recognise the wild type sequence, the mutant probe will recognise the known mutant sequence. In a heterozygote, both probes should work equally well. And remember, we're doing this by quantitative PCR mutation detection, and I'll explain how it, and I'll show you some traces from this experiment shortly. So just remember, the mutant and wild type probes differ by one nucleotide, one base difference over that about 12 nucleotide sequence. So that gives some degree of cross-reactivity, some degree of specificity, but it's not 100% perfect. So it's not that the mutant probe will not recognise the wild type and vice versa, it's just that the wild type probe will preferentially bind to wild type DNA. So here are some quantitative PCR based mutation detection amplification curves. So you've covered quantitative PCR in the past, you should have seen amplification curves. You should know that we draw a line across here at an arbitrary threshold and draw a line down to get the CT value. What we are doing here is analysing heterozygous DNA for a particular marker. We have got a probe that recognises wild type, we have a probe that recognises mutant, and in a heterozygous individual, both probes work effectively the same. we have pretty much the same CT value, we have the same total amount of fluorescence produced, so the mutant and the wild type probe are working with similar efficiency because there's equal amounts of mutant and wild type DNA. If we're dealing with a sample that's homozygous for allele 1, so in brown is allele 1, in orange is allele 2 in this particular example, what we can see is homozygous for allele 1, the probe for allele 1 has been detected earlier than allele 2, and the total amount of fluorescence is much larger for allele 1, and this is likely to occur because this sample is homozygous for allele 1. The allele 2 probe still recognises that DNA to some extent, but not as efficiently. And then oppositely, if we're homozygous for allele 2, the allele 2 probe will work more efficiently than the allele 1 probe. We'll get an earlier CT value for allele 2 because we're homozygous for allele 2, and we'll get more total fluorescence produced. This is a logarithmic scale, so this is tenfold difference there. What we can then do is take that information, a combination of the CT value, so is this CT value earlier than this one, and use that in combination with the total amount of DNA that is increasing per cycle, and if the CT value is earlier and the total amount of DNA or fluorescence is higher for a particular probe, then there is more of it, and we assume that that's homozygous. And then we can take that information and plot it on a 2x2 grid for allele 1 and allele 2. So these samples here are homozygous wild type. Allele 1 was wild type in this particular example. So they have earlier CT values and more fluorescence than allele 2. So this arbitrary scale here is saying this is strong amplification for allele 1 probe and weak amplification for allele 2 probe, so the samples are in this bottom right hand quadrant. Conversely, if the sample is homozygous mutant, which is picked up by the allele 2 probe, we have very strong amplification for allele 2, but very weak amplification for allele 1, so the samples are in this top left quadrant. And then any samples which are heterozygous run equally, there's equal amplification for allele 1 and allele 2, and we get these clustering as a line on this grid down here. And some samples amplify better than others, but allele 1 and allele 2 are roughly the same, therefore all the samples cluster as a line down there. If you do hundreds of samples, you tend to get this population extending as a line pointing down towards 0, and these samples extend as a line pointing towards 0, and these extend in line towards 0, and then the ones that are very close to 0, where there's limited amplification, they've got X's because they have failed. So doing this, we can quickly screen lots and lots of samples for a single nucleotide polymorphism or mutation and get a very accurate representation as to whether they're homozygous wild type, heterozygous, or mutant. Now, the situation where we might use this data here is from one of my master's labs that I run on molecular diagnostics. And what we're actually detecting here is a single nucleotide change in cutaneous melanoma for the gene BRAF. And the BRAF gene has a very specific mutation. It occurs at a known nucleotide and it changes amino acid 600 from a valine to a glutamic acid. So we call this mutation the V600E mutation. That's the change on the amino acid. The DNA change has got a different code, but we know exactly where it is, so we can design one probe that recognises the wild type sequence, one probe that recognises the mutant sequence, and then we can use this assay to screen melanoma samples to ask the question, is this mutation present? And this exact same mutation occurs in about half of cutaneous melanomas. And you might think, well, what's the relevance of that? Well, it's really important because there is a drug called vemurafenib, and vemurafenib is designed to only recognise the mutant oncogenes that have the V600E mutation. So if amino acid 600 is a valine, vemurafenib doesn't bind. If amino acid 600 is a glutamic acid, the drug binds and inhibits that BRAF protein and stops tumour cells from growing. We only give the drug to patients who've got this mutation, and one of the ways we identify these patients is we take their tumour DNA, extract the DNA, perform a qPCR based mutation detection assay and ask the question, are they mutant or not? So this is one of the ways in which you can do that. We could also sequence the DNA. So that's just putting it into context as to why we might do this. Okay, on this slide you've got a summary of everything I've covered in Lecture 1, MLPAs, and Lecture 2, single nucleotide polymorphisms and pathogenic mutation detection strategies by various different methods. So that's everything that we have covered.